Thank you for inviting me here to share my story with you. I'm working with company Bedrocan as the head of research and education, not development. That's a very different thing. And I'm going to share a little bit of 15 years of history of what we've done in the Dutch program. And to get right away started with it, why are we here? It's because we're trying to figure out what is medicinal cannabis. What happens when you take a plant with a long history, with lots of uses, lots of opinions, and you try to merge that with modern medicine as we know it. Pills and powders and injections and things like that. What does that look like? Well, the thing is that we don't know. It's a strange creature. And it's undefined, like this medieval beast that they thought really existed based on feathers and bones and fossils that they found from all over the place, and they put it together, but we have to figure it out. And usually when you try to know something about medicines, you do clinical trials, as been explained by the previous speaker. But if you don't know how to do clinical trials, or you simply don't have them, it's very common in medicine to look at traditional use. A lot of medicines that we're using today were discovered because we went into the rainforest in Brazil or Indonesia or somewhere else to learn what people have been figuring out over the centuries. Well, our people from the rainforest are now not living there, they're living in our own backyard, and cannabis patients from all over the world, they have a lot of information that could help us to design those clinical trials. And when I do a clinical trial, there's basically 1,000 things I have to choose. The administration form, the dose, the form of the cannabis, which compounds are supposed to be involved, what indication, and that's a hard choice because sometimes you just don't know yet. So let's look at cannabis patients to narrow down that search. Why not? So what I'm kind of suggesting is what I would call crowd researching. So yes, science has to make decisions and have to give numbers, because otherwise people like politicians and medical authorities don't know what to choose. But why can't patients inspire what we study in the first place? So I'm going to take you on a little tour, and I'm going to switch hands too, of what we have figured out over the last 15 years in Holland. And it starts with standardized cultivation. We do that here in Holland, in a little town in the north, not Amsterdam. It's the only town that anybody ever knows from Holland. We're not from Amsterdam. And we grow herbal cannabis, and we do it in a standardized way. I'm going to show you what that means. And we do it there with confidence and with a history because we're allowed to do it. Simple. Holland and Canada were the first two countries to allow it around 2001, 2003. So we'll be able to do everything under a legal umbrella. We're just scientists and growers and people doing our work. And this is how it starts, from clones. All plants are genetically the same. And then they go into a growing room, and it's a space about 10 by 10 meters, and all our spaces are in the same shape. And when the plants are harvested, they're turned upside down and they're dried under carefully set conditions, and then you have a bud, a flower top. And you have to cut off the leaves, and I see some people smile in the audience. They probably have done this work somewhere or other place. So supposedly it's fun if you don't do it for weeks at a time, and you get this dried cannabis flowers. This is somebody from the government. They come to take a sample. We don't test our own sample. You don't do that in medicine. They take it, send it to a lab, and then finally, if it's approved, you have a pharmaceutical quality cannabis flower, which is being distributed to patients in 5-gram jars. So if you need 50 grams, you don't get a huge bag, you get 10 of these guys. We do this in 2 locations. Some of them are dedicated to cultivation of the product for patients, other ones are for R&D. Those are 2 separate things by now. And over the years, since 2012, our production is going up very quickly in kilos per year. And that's because, I hope you can see this, around the world, things are happening. We're exporting. We have all kinds of clients in pharmaceutical industries, researchers from universities. We're growing in Canada and the Czech Republic by now. And the reason is that they like it standardized. If you are a PhD or you're doing a clinical trial and you're finding something exciting about your cannabis, you would like to order more. But you would like to order exactly the same. If you produce a patented drug based on cannabis, you want to make sure that in 15 years it's going to be legally available. So I'm going to have it delivered. If I order it, it's going to be the same stuff, and there's no trouble attached to it. That's why standardization is important. Patients are benefiting from the fact that they have the same result when they get the same dose every time. And we're also working on some product development ourselves. I'll show you more examples soon. But if you say it's pharmaceutical quality, that means I have to test something somewhere. And that testing has to be validated. You cannot just set up a lab and say, well, I can test your cannabis, just give me some and I'll give you a number, because is that number reliable? How am I supposed to know that it's the right number? And the basis, of course, is that cannabis is chemistry. It's a really beautiful plant with lots of stories, but basically it's a vesicle for chemicals. And we all know about THC, and there's a whole bunch of other chemicals floating around there. You can see them by name in all kinds of folders and publications, but those are just names, right? But in reality, they're molecules. You have to do something complicated in the lab to translate this into information that's useful. So if you want to do this, there's 100 cannabinoids, that's really great, but can you analyze all of them? Do you know what they look like? Can you quantify them? You need standards. And everybody working in a lab knows that the first thing you do when you're getting on a research project is you're going to order those things. Well, in 2003, when I started, there were no standards. So this is me. Around that time, that's my machine, the CPC in the background. I had to make standards because I couldn't order them. They didn't exist, they weren't delivered, they were too expensive, but basically we were on our own. And this is when we got started on this track of seeing cannabis as a therapeutic product that's basically chemistry and statistics. We have to figure out what works when, but we need hard data. By now we can analyze 48 components, and I would dare to say this is the widest range of cannabinoids and terpenes that can be measured anywhere. This took us years and years and years, and this is going to come back in a few slides. If you want to determine the quality of a cannabis plant, it's mostly, like I said, it's chemistry, right? You have to look for things that are supposed to be in there. You're also supposed to look at things that are not supposed to be in there, like heavy metals and pesticides, but you can't just use any method. You have to make sure that the method you're using is suitable for this sticky cannabis plant. Can I extract the microbes from a sticky cannabis plant to determine how many are there in the first place? It's not as simple as it sounds. But the most important thing here is that you want to know the cannabinoids and the terpenes. And these methods that we're using in Holland are all publicly available. You can look them up online, and they're validated according to pharmaceutical norms. So it's part of the scientific community, and if you look at those methods and say, I think you're doing something wrong over there, we'd love to hear, because this is how knowledge grows. If you can determine those cannabinoids, you can start to look at your plant and say, can I grow a standardized plant? Well, the rules are clear about that. Standardized doesn't mean it has to be 10.00% every single time. It's a plant, it's alive, it develops, so there's a little bit of variation allowed. Those are set by, for example, guidelines on herbal drugs, plus or minus 20%. And can we keep, for example, the THC in one of our varieties within those limits? Yes, that's possible. Can we do the same with the CBD in this plant? Once again, yeah, you can do that. When you control your growth, you can control the plant already in the flower stage. You don't have to fix anything while you're making a medicine out of it later. And most interesting, and we found this out last year, because now we control the analysis of all these compounds, we can see that even the terpene profile, and every one of those little peaks is one terpene that has been detected. And if you look at 5 different batches that have been harvested sequentially, it's virtually the same. So under the right conditions, you can almost completely control a plant. And actually, it's become our logo, because one day my colleague was showing this to some visitor, and he said, well, this is kind of how we do it. He looked down at the table, he said, hey, maybe that's something logo worthy. So we kept it. Well, now we're talking about cannabis as a medicine. It's been standardized, but the question is, what cannabis do we want? What about all these varieties? One of them is not the same as the other one, so how do we know what we're talking about? Well, this is where we start, and I usually refer to this as a Starbucks menu, because do you want a lot, medium, or small THC? Would you like to have some CBD to go with that or not? And would you like the sativa or the variety, or the indica variety? Soy milk, normal milk? And these numbers are pretty clear. THC is a certain percentage of the content, but then we're talking about sativa and indica. What does that mean? Well, we were struggling with that because we knew what patients meant with that, but how does it translate in the lab? We don't know how to quantify it, and we're kind of like feeling around in the dark. So this is what the internet has to say about it. Sativa does that, and indica does that. But where does this information come from? Is it true? Can you measure something? So let's try. Can we get our hands on hundreds of cannabis samples, extract them and test every single compound that we find, put them in large tables and try to find patterns? If there's a difference, we should be able to show it, right? So, of course, when you do that, you get tons and tons and tons of numbers. I've analyzed 460 cannabis strains for 48 components. That's about 25,000 numbers. And if I would project them on the wall, I can stare at it forever and I will never find a pattern. It's too complex. But the computer can, and you can ask the computer, can you see something that distinguishes sativa from indica? And actually, it's able to do that. This is the new paper is going to be published this month. So you can quantify differences. So now we have a reason to focus on one plant and not the other. We know what we're looking for. And just to simplify it, is nobody in this room, we compare it to a world map. Nobody knows all the 200 countries in the world, I think, but we all know that there are 7 continents. You probably know that. And that helps to understand how things are organized. So even if there's 1,000 varieties, great. But can we maybe bunch them up in 10 clusters? So when you have pain, you go to Europe. When you have epilepsy, you go to the United States. And when you can't sleep, you go to Australia. Something like that. Well, this is the map of all the 460 samples that I analyzed. And I will not go into detail what the map means, but if I remove the hemp samples, they're all together there in the middle. So they are actually chemically very simple. And I project the 6 cannabis strains from Bedrocan onto this map. And I see that there's 2 regions that are not covered by a product right now. And one of those clusters, I don't really know what's the common factor, but the other ones are all amnesia haze type samples, which are very common in Holland to find. Then we went to the patient organizations of the Netherlands and we asked them if there's one strain that you would bring to the market, and what would that be? Can you be as clear as possible? Because general terminology about cannabis doesn't always mean much to us, but we're trying to pinpoint something that's missing, and they all pointed towards amnesia haze. And then we thought, wow, these people knew nothing about this study, and just by tasting and talking and blogging and Facebooking, they figured this out. So call it the wisdom of the crowd, but we're going to introduce an amnesia haze later this year. But it's standardized, we know what's in it, and we can combine the 2 worlds of the lab and the patient. Well, all this information is going to a single point because, yes, we do need clinical proof. Because some people are very open to the therapeutic use, and I think indeed, like was just shown, that's a very important focus to have. Not everything has been or can be so easily clinically proven, but we do need it to put this discussion to an end and say, you know, it really does work. Like the report that was just shown for the UK, that's a list of clinical trials. You do need them to convince the last people who are not into it. But that clinical research can be patient-inspired. So what does that mean? Well, first question, what should I study? What is the indication that needs the most attention? It's pain. This is a survey we did among almost 1,000 patients, and pain in this survey in 2010, this is an international online survey, but any study and survey will tell you it's chronic pain every single time. Those people need cannabis. And if you look in the literature, yes, there's more and more patients being enrolled over the last 10 years, about 2,000 patients since 2005. The most important focus of those pain studies is neuropathic pain, so we're on the right track. But then if you see what they studied, what is the top 3 study medication? It's not herbal cannabis. It's oral pure THC, it's Nabilone, which is not a natural cannabinoid at all, or it's Sativex. So there's a gap here. People are doing one thing, scientists are studying another thing. What about administration forms? If I pick my indication and my cannabis, then how am I going to administer it? Well, we worked a lot on this device. A lot of the research going into this device came from my lab, but It's big, it's bulky, and it's expensive. So we asked a bunch of designers, can we come up with something smarter, smaller, slimmer? And you've seen a lot of vaporizers come out on the market in the last few years, and this is our idea. This is just a graphic. It exists for real right now. But why aren't we selling it yet? Because it needs to be studied. What happens when you change the cannabis dose? What happens when you suck harder? Not everybody has the same puff volume. What happens if you inhale 3 or 5 or 7 seconds? Does it make a difference for what you're getting into your lungs? Of course it does. So these kind of studies in the lab, they take a long time before you can even go to clinical trial. Another example is cannabis oil. I don't have to explain to you by now what that is. So the core of this discussion of this presentation is that can we have a system where we use the best of both worlds? If patients want to use herbal cannabis in different varieties and they want to inhale it or use it in an oil, is that really so much to ask? I think I've shown that that's a possibility, but you also have to keep in mind that modern medicine, it's 100 years old, and they have rules and regulations that make a lot of sense. So if you bring those two together in clinical trials, I think we're on the right track. And then finally, when you're working with information that you receive and ask from cannabis patients, and you try to do this in a collaborative way, you have to give something back. So all this information is available through education, courses, publications, and I think almost 98% of everything we've ever learned is in publications, so that science as a whole can pick it up as a tool and move on. So that was my goal today. Thank you.